Hello. The question that I'm going to answer today is focused on glucose metabolism in human cells. And the question asks us to consider what would happen to these cells if we treated them with a poison or a molecule that stopped the conversion of citrate to isocitrate in the citric acid cycle. The question further stipulates that we can assume that the citrate that would build up gets excreted from the mitochondria. So I'm going to answer this question assuming that we don't get a buildup of citrate, so we don't have any inhibition of this step that involves conversion of pyruvate and oxaloacetate into citrate. And the reason I'm making that assumption is because the question says that excess citrate gets exported from the mitochondria. So because this process is stopped here, anything upstream is going to be unaffected. So glycolysis is going to continue normally. So each molecule of glucose is going to go through the series of reactions of glycolysis, which, as we know, consume 2 ATP but produce 4 ATP. So there's a net gain of 2 ATP per glucose in the glycolysis pathway. We also produce 2 NADH for each molecule of glucose that goes through glycolysis and gets turned into pyruvate. Additionally, because I'm assuming that this reaction, the pyruvate dehydrogenation or decarboxylation reaction, can happen normally, that reaction takes pyruvate and oxaloacetate, converts it into citrate, and 1 NADH is produced as part of that. After that reaction, we hit this blockade, and that's where we stop. So the cell can't metabolize that glucose any further. So total, so far, we've produced 2 ATP and 3 NADH. Now, an important thing to remember is that this question doesn't tell us that the electron transport chain isn't functional. So we can assume that it is. So we've produced 3 NADH that can be used by the electron transport chain in the oxidative phosphorylation reaction to produce more ATP. Now, typically, we think that each electron pair from this NADH that's donated to the electron transport chain can produce somewhere between 2 and 3 ATP. And for purposes of this question, we're going to assume that the electron transport chain is operating at optimum efficiency and is producing 3 ATP per molecule of NADH. So this becomes 9 more ATP that we've produced from this one molecule of glucose. So in total, in this system, each molecule of glucose is going to be producing 11 ATP, 2 that come from glycolysis and 9 that come from oxidative phosphorylation based on the 3 NADH that are produced prior to the citrate reaction. Now, one thing to think about, because we're exporting excess citrate from the mitochondria, mitochondrial levels of oxaloacetate might start to drop eventually. And if that were to happen, then we could no longer convert pyruvate and oxaloacetate into citrate. And in that case, we would stop making this NADH here, which would mean this would go down to 6 ATP per molecule of glucose. I think that this question isn't asking for that level of detail, but it's something to think about that in real cells and real metabolism, anytime you tinker with any part of the tricarboxylic acid cycle, the whole cycle is going to be affected and eventually stop working.